Boy JOSHABRAHAM. We're back. We're back with the sequence analysis. It's been a while, people. It's been a while. This would be assignment number 9, and get idiot, I did not save the file. Turn the lights on. There we go. Yay! 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 Now UE. So, how is it? How is it this time? Exercise 10, copy number detection with hidden Markovnikov models. What the fuck? We got 4 questions. What? Submit a PDF file that contains your results and explanations plus Python code to obtain the results. Okay. Copy number background. Not necessary to solve the exercise. You are considering the problem to detect copy number variation of a gene in human genome. Copy number variation. Copy number variation are one of the main sources of genetic variation in mammalian genomes. A copy number variable region is a DNA segment larger than 1000 base pairs that appears in a different number of copies across the individuals. The usual case is that the diploid individual has 2 copies of each DNA segment. However, some DNA segments can appear 0 or 1 time, which we call a copy number deletion, and some DNA segments can occur multiple times, which we call an amplification. Many of these copy number variations are linked to genetic traits, phenotypes of even genetic diseases. Therefore, it is important to be able to detect the copy number of individuals. Detect the copy number of individuals. Okay. A highly simplified view of the copy number detection with microarray technique. For this exercise, we assume that a DNA of an individual has been measured by DNA microarray, such that the different DNA segments correspond to probes that react with the light intensity. However, a higher intensity indicates the presence of a DNA segment, lower intensity, the absence of a DNA segment. We assume that the probes of a segment with normal copy number emit an intensity with Gaussian distribution with a mean mu2 equals 7 and a standard deviation sigma 2 equals 1 ampere. 1 ampere? No, Armstrong. Segment with a copy number deletion corresponds to a probe with intensity of nu1 equals 5.5 and sigma 1 equals 2.0, and the segment with copy number amplification with intensity 8.0 and 1.5. Hint, note that these distributions are necessary to calculate PEXIUT from the lecture notes. We will consider 5 probes that are located in the consecutive DNA segments, such as the copy number in one segment depends on the copy number of the precursor segment. This means we will have all observation vectors, such as x equals, where each of these entries correspond to the measurement of one probe, and the probe marked in this order of the DNA. We will model the data arising from these DNA segments with hidden Markov model with 3 hidden states corresponding to the copy number status deletion, normal, and gain, where each state emits signals according to the above mentioned Gaussian normal distributions. The transition probabilities are given by where Aij corresponds to the probability of the state transits to state j, thus the row sum are 1. The initial state probabilities are okay. Black these girls, the crowds again. Black these girls, and Miss Mouse again. Short interpretation, how this ties to fixed algorithm. Oh, that's different. Let's just copy paste this and see what is this question about. Okay, from the looks of it, I need to understand. Observe data, light intensities from DNA microarray probes. Hidden truth, the copy number state of each DNA segment, which you cannot see directly. You want to infer the most likely copy number state sequence behind these. Oh, whoa. Good, you're right. This looks heavy, but the key point is exercise is about understanding, not coding. I'll strip it down to what you need to understand and where to start. What is the question actually? Oh shit, what is the question? Oh, it's just okay, it's not even the 0 points. Okay, okay. What is the problem really about? It's you were given observed data, light intensities from DNA microarray probes. Hidden truth. Shit, how long is the it's been going? It should be 1 hour. Come on. Come on, come on. 10 minutes, so I set a timer for 20 minutes. 20 Minuten. That is exactly what a hidden Markov model is for. Hidden truth, the copy number state of each DNA segment which, oh, you want to infer most likely copy number state sequence behind these observations. That is exactly what a hidden Markov model. Did I look into this Markov model? Please tell me I did. Go to playlist, sequence analysis, understand the fixed algorithm. Nope, I did not go for. Uh, I did go for Smith Waterman. Smith Waterman. Oh shit. Nope. So I don't know what Markov hidden Markov model is. What is the hidden states? These are three states. State meaning copy number. So the hidden sequences looks like what? But you don't know this. You only see the intensities. What are the observations? What the fuck? Okay. Okay. Looks like I don't understand anything. Should I watch a YouTube video explaining the Markov hidden Markov model? YouTube? Okay, I'll look into YouTube. Hidden Markov model. Why is it part 5? Let's see. Hello, people.
Normalized Nerd. Hello, people from the future. Welcome to Normalized Nerd. Today, I'm going to talk about hidden Markov models. This is a concept derived from our old friend Markov chains, and it is very useful in bioinformatics, natural language processing, speech recognition, and in many other domains. In this video, I will go through both intuition and the mathematics behind it. So make sure to watch this video till the end.
You know what?
I will also show you how Bayes' theorem helps us in this process.
I should
So if you find that
add my watermark.
please consider subscribing and hit the bell icon. That will help me a lot. So let's get started. Well, to understand hidden Markov models, obviously you need to know what a Markov chain really is. So I will highly recommend you to watch my previous videos before continuing with this one. Okay, let's start our discussion with our friend Jack, who lives in a hypothetical town. There exist only three kinds of weather in that town: rainy, cloudy, and sunny. On any given day, only one of them will occur. And the weather tomorrow depends only on today's weather. Yes, we can model this as a simple Markov chain. Let me add the state transitions. And here are the transition probabilities.
Mm-hmm.
Also assume that on any given day, Jack can have one of the two following moods, sad or happy.
Mm-hmm.
Moreover, his mood depends on the weather of that particular day. I'm going to represent it with red arrows. Here are the corresponding probabilities.
Mm-hmm.
So, according to our diagram, there's a 90% chance that Jack is sad given that it's a rainy day. Hopefully, it's clear to you too. Okay, so now our problem is we don't live in Jack's town, so we can't possibly know what's the weather on a particular day. However, we can contact Jack over the internet and get to know his mood. So, the states of the Markov chain are unknown or hidden from us, but we can observe some variables that are dependent on the states. And this, my friend, is called a hidden Markov model. In other words, a hidden Markov model consists of an ordinary Markov chain and a set of observed variables. Please note that Jack's mood, that is, the observed variable, depends only on today's weather, not on the previous day's mood.
Mm-hmm.
To make things more clear, I'm going to write the probabilities into a matrix.
Mm-hmm.
You are already familiar with the green matrix, that is the transition matrix.
Wait. So point 0.5, that's for rainy season to happen again. Point 0.4, that's for the normal day to switch to rainy season. And point 0, what the fuck is that? What? Point 3? Point 3, the rainy day, the chance of rainy day to go to normal day. Point 2 again. Again, we got a point 3. Is it the chance of sunny day to be a rainy day? Okay, that makes sense. But what's with the 0.0? What's with the 0.0? Huh? What's with that? What's with that shit? 0.2 rainy day to sunny day, 0.4 normal day to sunny day, 0.7 sunny day again sunny day. Okay, I get these three. But to the rainy day, again, rainy day, get that. Normal day to rainy day, I get that. What's this? What's this?
And the red one captures.
So if it's a rainy day, it's go okay. This is the sad. If it's a rainy day, you got 0.9% chance of being sad. If it's a normal day, 0.6. Sunny day, 0.2. Well, it's a rainy day, it's 0.1. Normal day, 0.4, 0.8. Okay, I just don't get this
corresponding to the observed variables, which is also known as the emission matrix. Let me add the indices to make it more interpretable.
Okay. Rainy day, 0.5. Rainy, sunny. Oh, there's no. Okay.
It's the correct time to pause and convince yourself that you have understood everything so far.
I did understand.
Okay, let's consider a particular scenario. We will look at 3 consecutive days. Suppose on the first day it was sunny and Jack was happy. Next day was cloudy and Jack was happy. And on the third day it was sunny again, but Jack was sad.
Oh.
Well, I know that we can't observe the hidden states, but just assume that this scenario happened. Trust me, analyzing this will help us a lot. Okay, the question I want to ask here is, what is the probability of this scenario occurring? More precisely, what is the joint probability of the observed mood sequence and the weather sequence? Pause here if you'd like to try.
What? Happy on the sunny day, that's 0.8. Happy on the normal day, that's 0.4. 0.8, is it plus, multiply? I don't know. And the sunny, normal, sunny. So, what,
Using the Markov property, we can compute this as a product of 6 terms. First, the probability of a sunny day, then the probability of happy mood given a sunny day,
probability of a happy mood given a sunny day, that's 0.8, okay.
then the transition probability from sunny to cloudy,
Now, probability of a cloudy day.
and so on. But how we got this?
What? The probability of a sunny day, being happy, sunny. What? X3 sun. Is it okay?
Terms. Please bear with me till the end to understand the underlying mathematics. We can fill the red terms from the emission matrix and the green ones from the transition matrix. But what about the first term? How can we find the probability of the first state? Yes, we need the stationary distribution of the Markov chain for the.
Wait. So y1 happy on a sunny day. That's 0.8. Okay, that's okay. Okay, I get it. Cloudy, sunny. What? Are we talking next day? I'm guessing we're talking about the next day. And the probability of being happy is gonna be a happy normal day. Why?
You can use the normalized left eigenvectors or repeated matrix multiplication to compute the stationary distribution. I have explained both of them in my previous videos. So, as you can see, the sunny state has the probability of 0.549.
You just multiplied pi with these? Is that how it worked? 0.5 into 1.57. No, it's not pi. How did you get it? How did you get it, bro?
We can compute the product. And the answer turns out to be 0.00391. Okay, let me hide the states now. So, we only have a sequence of the observed variable. Here I'm going to ask a more interesting question. What is the most likely weather sequence for this given mood sequence? There are
sunny, rainy. But sunny to rainy is not likely. So sunny, normal, rainy.
Any possible permutations, right? We can have rainy, cloudy, sunny.
Nope.
Cloudy, rainy, sunny.
Nope.
Rainy, cloudy, rainy.
No.
And so on.
It's sunny, cloudy, rainy.
To find the most likely sequence, we need to compute the probability corresponding to each of them and find the one with the maximum probability. We can calculate the probability just like we did in the last case. I wrote a Python script to do the computations and found that this weather sequence maximizes the joint probability.
Yeah, not gonna lie.
And the probability is 0.04105. If you are still watching this, I believe you would also like to know the formal mathematics behind this. So get ready for some symbols instead of emojis. I will represent the hidden states of our Markov chain by x and the observed variable by y. We can rewrite our problem like this. This simply means find that particular sequence of x for which the probability of x given y is maximum. Please note that in a hidden Markov model, we observe the sequence of y. That's why I have written x given y. Now, if you notice carefully, you will see there's no direct way to find this probability. Here comes the base theorem. By the way, I have a video explaining Bayes' theorem. You
Dude,
can watch that if you want.
what the fuck? You gotta explain every topic.
So by using Bayes'
Oh
theorem,
man.
we can rewrite this as the probability of y given x times the probability of x upon the probability of y. For all practical purposes, we can neglect the denominator. And the numerator is just the joint probability distribution of x and y. Now we are going to further simplify this. Let's take the first part, the probability of y given x. According to our assumption, yi depends only on xi. So we can write probability of y given x as a product. We can fill all these terms from our rate matrix. For the second term, probability of x, we must use the Markov property that says xi depends only on xi minus 1. So we can convert px into this product. Now there's one subtlety involved. For x0, we must use the stationary distribution vector as I already showed you earlier. Okay, so now replace these things into the above expression, and we have got the thing that we want to maximize. You can clearly see that this expression is just a product of two n terms. I hope now you fully understand the origin of those six terms in the example that I computed earlier. Isn't it super elegant? Well, I guess it is. I know it's not the easiest thing, so don't worry if you didn't get everything at once. You can always replay the video to remove your confusions. So that was all for this video, guys. Do comment below your thoughts and suggestions,
So,
and
a
subscription
hidden
will be a
Markov
major help to
model.
me. Goodbye, guys.
So, I kinda got some of those stuff, and this thing is basically what we have here. Where is it? Standard deviation. Yeah, this one, right? I think that's it. Transition probabilities A. Okay, B. Okay. Sub. Normalized nerd got a sub. Machine learning creative coding. The last video is 1 year ago. Bro, what? What happened, bro? 3 years ago, the last short, what happened to this guy? Okay, this guy's doing PhD, okay. That's good. That's good, huh? So, what the HMM in this assignment is modeling. These are the copy numbers, deletion normal amplification. At every DNA segment, the genome is one of these states, but you don't see it. Okay. These are measured intensities from the microarray. Observation, you do observe these. Each number is a light intensity from one probe. Now, I'm a bit. Oh, you could see my face. I'm a bit confused on what the probe is. It's 20 minutes. Should I do my workout? Let's see how bright it can get. Ah, what the? Ah, shit. That's too bright. Let me take a 5 minute break. It's too much. Okay, so we start a timer for 5 minutes. 5 minutes is over. Okay. Uh, I think you're crazy. I think you're crazy. I think you're crazy. Just like it, just like me. Ever since I was little, ever since I was. Ha ha ha, bless your soul. You think you're in control. Well, I think you're great. Well, I think you're crazy. Oh man, I can't do the high pitch, it's crazy. Could you? DNA segment in VetLab. DNA microarray works like this. You have a chip of thousands of short DNA sequences attached to it. You have a chip. I don't get that, friend. We don't get that here. We don't understand shit like those people. DNA segment probe. Uh-huh. I'll be right back, people. Ay kain, we bike. We bike, we bike. Oh. Right. You have a chip with thousands of short DNA sequences attached to it. Each of these short sequences is called a probe. Each probe corresponds to a specific location of the genome. Mhm. Each probe might target a small stretch of the probe to target the next stretch, etc. So, so each probe is a DNA sequence. Which is part of a larger sequence of an animal. Here we were genome. Each probe corresponds to a specific location of the genome. Now, you hybridize label DNA from a sample onto the chip. What? What? So is it the two sides or just one side? Give a shit. The microarray chips contain thousands of these probes, each designed to detect one specific small region. Don't come here. Okay. Uh, mhm. You hybridize labeled DNA from the sample onto the chip. The labeled DNA will stick to the complementary sequence of the probe. The more DNA is present, the more it binds. Uh, okay, could you explain this step in more detail? So I have understood that the chip is somewhat like a dish with a lot of DNA sequences. I'm not sure if they are uh, or long sequence. Like a dish with a lot of DNA sequences. I'm not sure if they're broken into small sequence or long sequence. And these probes that so I'm guessing the chip has DNA sequence that is separated with their 5 dash or at least uh, it's like the sticky ends as part of enzymes. I think that did a lot. I don't know. I tried some of the features from Apple Watch. So these are the things that I understood and my uh, unclear parts. So would you explain more on this? It's already 1 hour, okay. You're right. You're on the right track always. I'm on the right track every time. Let me clarify the width part, how microarray works. Step by step, you can connect it to the assignment. I don't need to connect it to the assignment. I need to know how do we get from real world to computers? How is it connected? How did a drop of blood or anything organic is in the computer in form of sequences? That's the part I'm curious. Each probe, these are the probes. Each probe is short and represents a small segment of a gene or genomic range. Mhm. These probes are fixed in place. They are single-stranded DNA. They can bind complementary sequences. Okay. Dude, I can literally feel my attention span kind of giving up. My attention span is kind of starting to give up. Come on, bro. Don't give up. Think of it as a solid surface like glass that has thousands of very short DNA sequences attached to a specific location. These are the probes. Each probe is a short, represents a small segment of the gene or genomic region. These probes are fixed into place. They are single-stranded DNA, so they can bind complete complementary sequences to sample DNA. You extract sample from your you extract DNA from your sample. The DNA is usually fragmented into smaller pieces because the full genome is too long to work with. Then the DNA is labeled example fluorescent tag so you detect the binding hybridization. The labeled DNA factors are washed over the chip. Each fragment will bind only to probes that are complementary. This is like a lock and key mechanism. If okay, the strength of the binding reflects how a DNA segment is present in the sample. For example, if a segment has normal copy number, they get a standard intensity. If the segment is deleted, less DNA binds, lower intensity. If a segment is amplified, more DNA binds, higher intensity. Misunderstandings cleared. The chip does not contain long genomic sequences, it only has a short probe that's fixed. The orientation matters for complementary, but they don't have a separate strand floating around like sticky ends. Each probe is designed to bind to its complementary fragment. Chip is like the map with thousands of bait sequences, and label DNA sticks only to the matching spots. The intensity at the probe tells you abundance of DNA Okay, you know what, YouTube? I'm gonna end the stream. I think my mind is almost there, but we will be back in strong. Peace.